So welcome to Daylight Savings Time. Everyone awake and early this morning. Good thing. Let's see, is this actually working? There we go. Quick reminder, tomorrow at noon, research round seminar by Nacho. Actually, I think you can put it on the slide here. De la Higuera, postdoc who's working with me, talking about some of the stuff that he's done in the past and some of the stuff that we're doing now in my lab. Next week is the final. It's at 8 o'clock, yes, 8 o'clock on Tuesday, which is the regular exam time for classes that meet Monday, Wednesday, Friday at 9. So 8 o'clock on Tuesday in here. As always with my exams, 50 multiple choice questions, not cumulative per se, but I will assume that you know some of the material that we've talked about beforehand. So another thing is, last time I said that we would have a guest lecturer on Wednesday. Unfortunately, that guest lecturer has a very small child who is not sleeping very much, and so I will be lecturing on Wednesday instead of my graduate student. So I'm actually kind of looking forward to it because CRISPR and RNA regulation is actually really fun stuff. So that should be, at least from my point of view, a lot of fun. So let's switch over and continue. Oh, are there questions again about that? Yeah.
I remember we took a quiz at the beginning of the term.
Yeah.
What was the point of that?
What was the point of the quiz at the beginning? That you got extra points. That was the point of that quiz.
Oh, I was just curious.
Yeah.
I didn't see it.
Yeah.
And did we
Yeah.
take a follow-up?
Pardon? I can offer you a follow-up after the end of the term. So sort of the idea for me was really to take a look and see what people knew and what they didn't. And now at the end of the term, hopefully you know all of that stuff, right? So maybe I could, if you like, I can share all the questions like after the exam. So yeah, you can now answer all of them because you've been ridiculously helped in your education by this course or something. Yeah, but no, the other thing is I have not added those points yet. So I will add those points. Those points just had to get added to clicker questions at the very end. OK, are there questions, comments, worries? Again, the material will just be through Wednesday's lecture. It'll be on the final. Friday, there'll be a review. There will not be any clicker questions on Friday. If people would want extra office hours, email me, and we can try and set up some more, maybe potentially even on Monday next week, if people don't have other exams that they're doing. OK, so today we're going to finish up talking about transcriptional regulation and then move on into post-transcriptional regulation. And when I say post-transcriptional regulation, it's post-transcriptional initiation regulation. Today, wanted to spend some time talking about some regulatory circuits, X inactivation, DNA methylation, imprinting, and sort of an overview of epigenetics, and again, very much an overview of epigenetics. So to start out with, what we finished with last time was phage lambda, exactly how this works. Take my virology course next term, and we'll talk much more about the details. But the basic sort of transcriptional interesting issue here is that when lambda, this bacterial virus, infects a bacterium, in this case E. coli, it has to make a decision. And that decision is, do I blast apart the E. coli and make a whole bunch of new viruses, or more specifically virions, or do I just hang out inside the cell and wait until the going gets bad? And that decision-making process, this is a bacterial virus. It's got 50,000, or actually even smaller than that, about 44,000 base pairs in it. How does it make that decision? It certainly doesn't have any kind of nervous system. And so that really is based on what's been called the genetic switch. And so a decision-making process, so signals that come into the cell, and those signals are being sensed by the bacterial virus infection, and then it makes a decision. And then once it's made that decision, it actually continues with that decision. And so here, if it decides that it's going to go what's called the lytic cycle, where it blasts open the cell and makes lots of viruses, once it makes its decision, it sticks with that decision. Same thing here, and particularly important here, if it decides to integrate in the genome, it stays with that decision over time. And so how does that work? Well, it turns out that it's actually a pretty straightforward mechanism. Exactly how the sensing works, again, we'll talk about next term. But there are really two kinds of states that this virus-infected cell can be in. It can be in a state where it just hangs out, and again, multiple rounds of replication. And that state is what's called the prophage state. The virus genome is integrated into the host genome. We talked a little bit about the lambda integrase weeks and weeks and weeks ago. One of these site-specific recombinases. So it takes the viral genome and puts it into the host genome. It can also chop it back out. Once it's in the host genome, it makes this protein that we've talked about already called the lambda repressor. And this lambda repressor does first represses genes, and these are the genes here, the so-called CRO genes, or the control of repressor and other genes. They're turned off, and that's because this repressor sits down on the promoter, doesn't allow the RNA polymerase to bind. But at the same time, the lambda repressor, as we've talked about before, also serves as an activator of its own gene. And so the lambda repressor sitting here blocks these genes and causes the expression of other genes. And what this does, it gives you a positive feedback loop. So if you're making the lambda repressor, it makes more of the lambda repressor, which makes more of the lambda repressor, which makes more of the lambda repressor. And so in that way, you are keeping one particular state. And so the switch has been made. You're going into this lysogenic state where you're hanging out. That's being maintained. On the other hand, sometimes the virus decides it wants to go this way. And this is the lysis pathway. And it turns out this is regulated in exactly the same way, only a different protein in a different place. So the CRO protein, control of regulator and other, also binds to this operator sequence, but it binds to the opposite end. So now it blocks the binding of the polymerase from making this lambda repressor gene and stimulates expression of the CRO gene, and you continue to make CRO. And if you make CRO, then that stimulates the expression of CRO, which stimulates the expression of CRO, et cetera. And all the time, this lambda C1 is turned off. So this is a classic positive feedback loop, and this is how cell memory very often works. You turn on a gene at some point due to some kind of signal, and again, we'll talk about that for lambda next time, next term in virology. But once that signal's been made, you maintain that signal. And this also seems to happen in lots of development in humans and other animals, etc. And that's just cartooned here. Have a signal right here which turns on the expression of some kind of protein, and in this case, it's an activator of its own gene, and this activator protein turns on its own gene. But the really nice thing is when you undergo cell division, you're going to still have some of this A protein around. And in the new cell, that'll make more of the A protein. In the other daughter cell, that A protein is going to link into making more of that A protein. So this is, again, a very classic way that cells can remember things. Turn it on once, and then as long as it stimulates its own production, you will continue to keep that going. Now, this is what happens for activators. You can also do very similar things for repressors. And so if you make a gene which represses its own production, what that does is what's also called a negative feedback loop. So here we have the geneticists like to have these bars which represent blocking some kind of activity, in this case transcription, and the arrows being activation. So that was their positive feedback loop. The negative feedback loop, and then there are lots of combinations of positives and negatives that you can do, and electrical engineers know all about this stuff, probably more so than I do. But the basic idea here is that combinations of turning things off and turning things on, and the fact that when we're doing this, you have a time delay. So when you turn on transcription, first you've got to transcribe that gene. That messenger RNA has to get, in the eukaryotic case, processed. In bacteria, it's getting translated. In eukaryotes, it's also going to have to be translated. That's a time lag. And then once you've made that protein, it can go back and do whatever it was doing in the first place. And so together with these activations and repressions and a time lag, you can actually have really complicated systems, and the book gets into this in a huge way. This is sort of how the engineers start to think about biology. And I don't know, how many made it to ladies' lab night? Any of you make it to ladies' lab night over the weekend? That was really fun, and one of my ex-students talked about some systems biology, and so how people are now the engineers are trying to engineer how you can get bacteria or other cells to do what you want them to do, but also to predict how some of these cells are likely to function. And it's really a combination of all of these different kinds of things here. And so just looking at a few of these gene regulatory circuits, I already talked about positive feedback, and again, the example of lambda, activators which activate its own gene. But negative feedback is also really nice because, again, given this time delay, you make enough of a protein to get to a certain point, and then you shut down overproduction of that protein. And it turns out in the lambda repressor case, you also have one of these as well. It gets to a certain point, and then it stops. And having that kind of control is also really important. You don't want to have this runaway train kind of effect. So having some way of shutting things off is really classic a negative feedback loop. And then just by having positive and negative regulatory systems that are going to regulate each other, you can turn things off, turn things on, sort of a flip-flop switch. And in fact, a lot of circadian rhythm, why it was a pain to get up an hour earlier this morning, has to do with the rhythm of genes getting turned on and turned off. Our circadian rhythm has to do with regulation of different genes, and in fact, people have artificially put a bunch of repressor proteins into cells and see that they have a circadian rhythm. And again, through this regulatory process. And then the last one is this, what's called a feed-forward process. And this is literally just two activator proteins right here, activator protein A and activator protein B. You only get an output here if you have active A and active B. And because of this time delay, you'll only end up with a full output if you have this input happening for a long time. So you have a signal that happens for a long time. This is just shown here. So you have a short signal. This is a little blip. Then you turn on A, but you don't have enough time to turn on B as well to get an output, and then A is gone. However, if your input lasts for a long time, you have enough time to make A and enough time for A to make B, and you end up with this output process. So, and this is just literally, it's a kind of a rheostat. You turn things up to a certain level, and then you'll get a response, as opposed to just having some kind of noise, a little bit of activity, which is not going to be followed along. And this is, again, very much a basis for thinking about how you can understand how all of these things are starting to fit together. And a lot of, again, the work done on this comes from things like electrical engineering and some systems engineering processes, looking at all of these interactions and trying to understand cells. We're getting pretty good at bacteria. Beyond that, they start to get a little bit too more complicated and more of a change to think about. But it's literally just combinations of activators and repressors. That's partly why we spent so much time talking about activators and repressors so far, is that that's where a lot of this regulation is happening, and through that you can get all of these interesting regulatory circuits. Quick aside here, I wanted to talk about not so much the activators and repressors per se, but how we can look at some extra changes that are happening above and beyond those activator and repressor proteins. And one of the best examples of that is the case of X inactivation. So actually probably more than half of us here have 2 X chromosomes. Some of us only have 1. But one of those X chromosomes has been inactivated in most of our cells, because having 2 active X chromosomes, with a few exceptions, generally is pretty problematic. And so even though females, in terms of their genetic components, actually have 2, you can't be expressing from both at the same time. So this is a process called dosage compensation. It's a little bit different in mammals than in some other, say mammals other than other animals. And is literally, again, just turning off one of these two X chromosomes, either the paternal X chromosome or the maternal X chromosome. In early embryos, actually, both of them are active. And then at a particular point, one of them, and it seems to be pretty random, gets inactivated. How this happens has to do with the ratio of X to autosomes. How that's measured is a really fascinating process that we're not going to get into. But the outcome of this is that in female cells, you have these things called bar bodies. And all that a bar body is an inactivated X chromosome. And that inactivated X chromosome, the reason it's called a bar body is because it's really compacted. And that compaction is probably the best understood example of heterochromatin. Really, really heavily compacted. And you can literally see these in stains of nuclei, where you have a spot, which is where you have a compacted X chromosome. Classic example of this is what are called the calico cats. Calico cats have all of these different colorations. The reasons they have different colorations is because in the females, one or other of the X chromosomes has been inactivated. And they're random all over the cat. There's no such thing as a multicolored calico male because they've only got one of their X chromosomes. That X chromosome is not being inactivated. What this means is that actually all females, genetically speaking, are mosaics because some of their X chromosomes are going to be turned on in some cells and some are going to be turned on in other cells. And that seems to happen pretty early in development. However, once that happens, once you have one of these that's been activated, say in this lineage, the maternal X which has been inactivated, that's actually maintained, that you have heterochromatin that's being maintained. We've talked a little bit about heterochromatin being maintained already once you have the modifications, and a lot of the modifications here, as we'll see, happen to do with histones. Those histone modifications, there's a histone read-writer complex that binds to it and then also modifies the histones that are right next to it. But the big question is, and this has only been found out about 10 years ago, is how that first step actually happens. So the way that first step actually happens is, again, one of the two X chromosomes is going to become inactivated. One of them is going to become a bar body. How does that happen? There's this non-coding RNA, and this is actually one of the very first non-coding RNAs that wasn't known to be a ribosomal RNA or a tRNA that has a particular activity. And so this XIST RNA, pardon me, is expressed, i.e. it's transcribed, on one, but only one of the chromosomes. And again, this seems to be pretty random. But once it's been expressed, what happens is this RNA actually starts to coat the chromosome right next to where it's being made. And that process of binding to and coating this X chromosome causes then the binding of proteins. These proteins are specifically proteins that will remove acetyl groups from histones. So histone deacetylases. So you take off all of the acetyl groups that were present on the lysine side chains of those histones, and then they get methylated. You also have a new histone that gets added, and then eventually what happens is that DNA also gets methylated. And we'll talk about DNA methylation a little bit later on. And it's that combination of all of these things, the coating of the RNA, the lack of acetyl groups on histones, presence of methyl groups on histones, and then finally the methylation of DNA, which leads to a completely inactivated and, of course, we saw from the bar bodies, a completely compacted X chromosome. And so that is now compacted. And again, as soon as this gets replicated, yes, it's got to decondense in order to replicate, but you still have the alternative histones. You still have the modified histones that will lead to that X chromosome continuing to be the one which is inactivated. So what about DNA methylation? Where does this whole DNA methylation come from? We talked about DNA methylation, we talked about DNA damage, but this is not from DNA damage. It's actually a regulatory mechanism. And we also talked about DNA methylation in bacteria. That's to protect against restriction endonucleases and also to give you licensing for when you're getting replication. Here, this is in eukaryotic systems. You have a methylase, and this methylase will bind to heterodimeric DNA that has a methyl group on one strand, and it will just methylate the group on the other strand. This particular methylase binds to CGs, so also known as CPG. Why they call it CPG, I don't know, but it's just a cytosine guanine from 5' to 3', and of course, the opposite strand is going to be cytosine and guanine from 3' to 5'. But the CPG is basically that there's a dinucleotide right next to each other, again, in the context of the rest of your DNA. If you have a methylated group on one strand, that will get methylated on the other strand. Where is this methylation? It's 5-methylcytosine. 5-methylcytosine, great, modified, doesn't change any of the base pairing, because of course the base pairing is happening on this side of your cytosine. What's the big problem with this? Big problem with 5-methylcytosine. Not so much the way that other molecules interact with it. What happens if you deaminate this 5-methylcytosine? What do you end up with? You end up with uracil, exactly. Is uracil base pair the same way cytosine does? No. So classic way that mutations are generated is you actually end up deaminating this cytosine. Deamination of this cytosine would give you uracil. Deamination of this gives you what? Thymidine. So there's no repair mechanism. If you deaminate cytosine, you've got uracil. You've got a repair mechanism. You've got uracil DNA glycosylase. If you had a thymidine DNA glycosylase, you'd be chopping out all your thymidine in the DNA. That would not be good. So this is a modification, which turns out to be very useful because there are DNA binding proteins that interact with it and help to silence DNA. But the danger is you have deamination, and then you end up with mutations that happen in this particular region. So the main thing that DNA methylation is used for is, again, gene silencing. And this is actually a lot like the process that's going to happen in the inactivation of the X chromosome, only it's the RNA here rather than a protein, gene regulatory protein. Modification of your histone side chains, methylation, your read-writer complex, which leads to methylation of all of these. Once you have methylation of your histones, then you recruit a DNA methylase, which is what these little dots down here are supposed to be, and you'll have the DNA, or say the methylated DNA binding protein, which will also bind to this and lead to a complete compaction of this piece of DNA. Also, this is known as gene silencing, and it's not just the methylation, but it's the methylation plus the binding proteins, which means that an active DNA versus a DNA, you get about a 1,000,000-fold reduction in the expression of genes that are in a silenced part of DNA as opposed to a non-silenced part of DNA. So really, really turn off the expression of genes. The best you can do in bacteria is only about a 1,000-fold reduction in terms of genes being on or genes being off. So 1,000,000-fold reduction makes a big difference. And it turns out that this is really important in our genome, and in fact in most mammalian genomes, because we've got all these retrotransposons in our genome. Retrotransposons in the genome, if they get expressed, they'll make the endonuclease reverse transcriptase. They'll jump around. They may jump into a gene that they're not supposed to be in and cause lots of mutations. And so this gene silencing process seems to also be really, really important for shutting down the activity of transposons. And we'll see also from an RNA level how that works a little bit later on. So methylation, DNA methylation is great for gene silencing, as I forgot to mention here, so I put my notes at the bottom. Turns out that if you lack these methyl groups, you have all kinds of problems in development. In fact, a lot of this work has been done in fruit flies. So you do have to have this methylation, and sometimes this methylation is also reversible. You can have demethylases that will happen, and we'll talk about demethylation a little bit later on when we talk about imprinting. So methylated DNA comes and goes. Once it's made, happens on these CPG islands and is mostly important for repressing activity of that particular part of the chromosome. One thing that was actually a little surprising when the first human genome sequence was done, almost 20 years ago actually, the first release of the human genome sequence came out, is that there are some areas of the genome where there are lots of these CG sequences. And these particular CG sequences, again, this was just in sequencing the genome, they said, wow, there's a bunch of places we get a whole bunch of CGs, but lots of the genome where you don't have CGs. And the question sort of arose, well, why would you have these? And these CG sequences, people also call them islands because they're spread out, they were not randomly distributed in the genome. Thousands of nucleotides, between 1,000 and 2,000 nucleotides, and they turned out to be right next to promoters. And in retrospect, people have gone back and said, oh, well, finding these CPG islands is great because they are right next to promoters. So the next question is, why are they next to promoters? Why do you have these CPG islands right next to promoters and not in all the rest of the genome? Why might that be? Well, what were we just talking about in terms of what happens if you have a methylated C and you have DNA damage? What happens? Methylated 5-methylcytosine becomes a what? Thymidine. What does that do? It's not getting repaired. What happens? You have a mutation, right? So you end up with a T after that gets replicated and an A on the opposite strand. So if you have a region where you can have mutations and they are maintained, then it's not a problem if you go from C to T, right? But if you're regions where you absolutely have to have that particular sequence, say in a region of the genome where you have gene regulatory proteins binding, where you have pre-initiation complexes binding, then maybe if there were mutations that happened there, then they wouldn't be maintained. That would be something that would be negatively selected for over evolutionary time. So that's why people think that these CPG islands are located next to promoters, and they're in these regions of the genome where you often have genes that are being expressed all of the time. And so here's, I think, the best example of that down here at the bottom, ribosomal protein genes. You need to make ribosomal protein genes in all cells all the time, and this has this really nice CPG island right next to it. So you can't modify that, because if you did, you'd have problems getting transcription and making that ribosomal RNA gene, and you need ribosomal RNA genes, or ribosomal protein genes in this particular case. But if you had any kind of silencing of that region, you would also be having problems. You silence that gene, you're not going to get expression. You can have mutations in that particular gene. So those seem to be the reasons why we have CPG islands. It's from an evolutionary point of view, but also you don't want to be silencing any of these genes. It turns out to be a really great tool. You can just literally go through the genome and say, hey, there's a bunch of CGs right here. That means I probably have an expressed gene right next to it, even if you have no idea what that sequence might happen to be. Questions on this? Means it's time to grab your clickers, right? Everybody's clear on CPG islands and everything else? Or not, as the case may be. Oh, actually, I should turn on our clickers. Well, we can take a quick look at the clicker question, and I will now turn on the clickers. So continue to look at this, and I will turn on the clicker software.
So do you turn it on when you need it now?
We'll see. That may or may not make a difference. Actually, I forgot this morning, so. So here we are. Why is it thought the CPG islands are located in promoters? Because promoters are preferentially methylated, a random chance. Because promoters are protected from methylation by DNA binding proteins. Because promoters are protected from methylation by barrier sequence binding proteins. Because promoters are protected from methylation by insulator binding proteins. Look at the whole screen now. Ten, 5. Okay, we're still not quite where I would like people to be, so tell your neighbors what you chose and why. Go again? Go. Ten, 5. Is that Stedman number C again? Well, it's definitely not because they're preferentially methylated, because you find that you wouldn't find them there if they were, because you'd end up with deamination and they'd turn out to be T's. Random chance, they would be randomly all over the place and you wouldn't see them. Barrier sequences and insulator binding sequences. Barrier sequences are heterochromatin. You're not going to have heterochromatin right at a promoter. Insulator binding is also where you're separating regions of the genome from regulation from other parts. So you're really actually kind of only left with C, but it's also Y, because if you had methylation, your DNA binding proteins, they're also going to be binding, and I forgot to mention this, we talked about where the methylation is happening in the cytosine. It's not where you have base pairing interactions, but it's in the major group of DNA. And so that's where almost all of our sequence-specific DNA binding proteins are. So yes, we have C. So there's another place where you have Okay, this is where we're going to have our issues again. Maybe not. DNA binding proteins interacting with methylated DNA. So usually, again, methylated DNA is going to lead to gene silencing because you have read-writer complexes. It's also going to interact with your histones, just also interacting with DNA as well. But there also are these DNA methylations which are inherited in very weird ways. And this is what's called genomic imprinting. So in genomic imprinting, what happens is you have methylation that happens on one of the two copies of your chromosome, so one of the homologous chromosomes, and that chromosome turns out to be always from one parent. So in this case, always from the father. You can also have genomic imprinting that happens from the mother. And what that means is one of the alleles has methylation and the other one does not. And this is another example, like we had with gene conversions before, where you don't have regular Mendelian inheritance. That process, again, it's methylation, and it's methylation in one of the sets of chromosomes that you get from either your mother or from your father. And so how this works is you have methylation, okay, methylation again, in this case in the paternal chromosomes, and then you have removal of imprinting in these germ cells. And you have meiosis. So meiosis, again, you get eggs, so you've gone from diploid to haploid. And then, in this case, in the male cells, you have imprinting of both of the chromosomes. Offspring here, their paternal chromosome has been imprinted. Turns out that this imprinting is an absolutely critical process in brain development and a number of other genes where it turns out that just having one copy now of autosomal chromosomes as opposed to the sex chromosomes is really important. So just one of those, an expression from just one of them. Turns out this is pretty much only present in mammals, and it's only about 100 genes of the 20,000 that we have. But they are really pretty important. So there's a couple of ways that these things can work. One of them is just as you would expect. If you have a methylated chromosome, you won't have proteins that bind to it. But it turns out that methylation, and people have looked at this in terms of a couple of different genes that are known to be imprinted, can also serve to activate genes. And so the best known of these is a gene called IGF-2, insulin growth factor. Again, growth factor important for development. Normally, in the case of a non-imprinted gene, you don't have activation of this gene because there's an insulator between the regulatory sequences and the gene which is being regulated. And that insulator sequence, we've talked about insulators before. How does an insulator work? An insulator works by an insulator binding protein that interacts with that, and usually then with another insulator, which we'll use to block off this particular gene. Well, if you methylate that insulator element, which is in imprinted systems, and in this case in the paternal chromosome, then this regulatory sequence can get past that insulator and express this particular gene. And again, this is really important in fetal development. Turns out you can also have methylation which can change which genes are actually being made. And in this case, there's a gene which is normally, the name of this gene is not important. The name of this gene, also IGF-2, is not important. It's not going to be on the exam. But you have a messenger RNA which is made and turned into a protein if this gene is methylated. Now, the methylation is right in the middle of that particular gene. However, if you have non-methylated here, there's a promoter which otherwise would not be used, which makes this long non-coding RNA. And we're going to talk a lot more about long non-coding RNAs later. But what happens with this long non-coding RNA is the expression of this will then block the expression of this particular gene, and you don't make this protein. So it's here you have a methylated gene where you're making a protein, but the non-methylated form leads to blocking of the expression of that gene. And so these are the examples I was saying that most of the time, DNA methylation, silencing, shutting down, compressing this particular part of the genome. But in a couple of other cases, and again, these imprinting cases in particular, you can have methylation that leads to activation of genes, or methylation in this case, with a lack of methylation, leads to non-expression of one particular gene. So imprinting can both deactivate and activate different genes. I'm going to finish up talking about transcriptional regulation by a very, 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 very brief introduction into epigenetics. I've actually talked about a couple of these things already. So most people, when they think about epigenetic inheritance, they think about histone modification. So we talked about histone modification. These are our read-writer complexes. So read-writer complexes, you have modification of a histone. The reader complex is also connected to the writer complex. This will give you an inherited chromosome state, or the chromatin state. Again, all of this epigenetic inheritance is in the absence of any kind of genetic change, no change in the nucleotide sequence here, but it's something that can be inherited in new daughter cells. DNA methylation also can be inherited. Again, it's through these read-writer complexes. So if you have methylated DNA here, you will also get methylated DNA here, and because of the maintenance methyltransferase, as soon as you separate those two strands, the new strand will end up being methylated as well. And this also happens in bacterial systems. So this is a change in what's happening with your DNA, but not a change in terms of the genetic sequence change. You can also have epigenetic mechanisms that happen through these different feedback loops, and this could be a positive feedback loop, could be a negative feedback loop. Anytime you have a protein that's present inside a cell, when you have cell division, you're going to end up with those proteins. And so here you have a protein that's been turned on, activates its own gene. As soon as this cell divides, there'll be some of that protein in there, which will also turn on this gene, and that would have been because you had a signal at some point where protein A was made. One that we haven't really talked about yet, but is also one of these epigenetic states, is if you have a protein that has two different kinds of folds, and this is best known in the prion proteins, also probably true for Alzheimer's and Parkinson's, some of these neurodegenerative diseases. You have differently folded proteins, and those differently folded proteins can actually stimulate different folding of that same protein. And so here we have a misfolded or a differently folded protein. The presence of a misfolded protein, even if you have some of the regularly folded protein, it will then get converted into this misfolded protein. And so again, this is a inheritable state. So all four of these are ways that you can have inheritance that is not based on some kind of genetic change, which is why it's called an epigenetic change. All clear on epigenetics? Yes, good, no? Not going to ask you a clicker question quite yet. Review transcriptional regulation. So we've talked about this for most of the last 4 lectures, which means it's going to be a lot of this stuff on the exam. DNA binding proteins, helix turn helices, classic motif that you have for binding of bacterial proteins. Almost all of our DNA binding proteins are going to be interacting with alpha helices sitting in the major groove of DNA, and can bind, of course, in the absence of any kind of the strands coming apart. Just the mostly hydrogen bond interactions that are happening in the major groove of DNA. We talked a little bit about those methods that you can use for that, band shift methods, DNA footprinting methods, and chromatin immunoprecipitation methods, as well as purification methods. The mechanisms on how these regulators work, bacteria, pretty simple, but it's not just repressors, as even though Jacob Monod thought that was the case. It's repressors and activators, but you can have repressors that will block expression in the presence of a ligand. You can also have repressors that will fall off of the DNA in the presence of a ligand, depending on what you need to regulate after that. And repressors in bacteria are almost always going to be overlapping with the binding of the RNA polymerase. Activators are going to be right next to the RNA polymerase, with that one exception that we talked about that I spent 6 years of my life working on, which is NTRC, which interacts at a distance through looping, and that's how most of the eukaryotic activators work, by binding to enhancer sequences. An enhancer binding protein is an activator that then can loop around through space and interact with proteins at your pre-initiation complex. Repressors can act just like you have repressors acting in bacterial systems, but much more often they're interacting with the activator itself, either by blocking the activation part of your activator, which is the sticky part that helps to bring all the promoters together, but also in terms of interacting with chromatin. So chromatin, and particularly chromatin remodeling complexes, histone modification enzymes. This is something that you don't have in bacteria, at least not the histone-based chromatin. And activators are going to lead to modification of histones that allow the transcriptional apparatus to bind, and repressors are going to be interacting with histones in such a way that it will stop the pre-initiation complex from associating with the promoter. In eukaryotic systems, you almost always have both repressors and activators working in concert at particular promoters. The example we talked about there was the even skipped process. Regulatory circuits where you have one gene leading to expression of another gene. Today we talked about the X inactivation process where you have a non-coding RNA that leads to lots of chromatin modification. Also, the final step is sort of the DNA methylation process, which usually again leads to suppression of transcription, but in the case of imprinting can also lead to some kinds of activation, and then finally some epigenetic processes. So we have questions on transcriptional regulation at this point. Okay, so let's talk about what happens after you have transcriptional initiation. So people always talk about this post-transcriptional regulation. I like to say this is really post-transcriptional initiation regulation. So it's before you actually get to your final protein synthesis down here at the bottom. So you can start your transcription. In some cases, you stop transcription before you get to the end of your transcript. Capping is regulated. Splicing is regulated, and we'll talk about a couple of examples of splicing regulation. One of the neat things that happens is also you have RNA editing that can take place. So once you have transcribed a particular gene, of course, we all know our genetic code. No, I'm not going to ask you all the details of the genetic code, but the genetic code is known, and we can go from a protein sequence to a nucleic acid sequence to a protein sequence. But if that nucleic acid sequence actually gets modified, then you can end up with a different protein. And it turns out this happens not frequently, but occasionally you'll also have some RNA editing. Turns out there's more and more that people are understanding about RNA editing as time goes by here. But then once you've made your messenger RNA, at least in eukaryotic cells, it's got to get out. And so regulation of nuclear export is also an issue, determines the amount of final protein that you have. Once you get out, where your RNA happens to be. And this turns out to be exactly what happens with getting those gradients of the regulatory factors that you see in Drosophila embryos. It's about where that RNA is inside the embryo that gives you a particular gradient, which will turn on some of these genes later. Translation is regulated. When you start, where you start. And even in some cases, you have recoding processes. We talked about this a little bit already. Frame shifting that can take place, where you have a RNA which codes for one protein in one reading frame, but a slightly different protein in another reading frame. And then how much of the final protein you get is going to depend on how much of the RNA you still have around and how long you have it around for. So I'll talk about a couple of examples of this. This one, I've actually scribbled out the C-terminal domain of the RNA polymerase here because it's actually a bacterial system that this is used for. These are what are called ribose switches. So what's a ribose switch? Ribose switch is a sequence of RNA, and that sequence of RNA can form a structure. And this structure, just because of base pairing interactions that form in the RNA, also known as an aptamer. All of this sequence right here is an aptamer. And this sequence, which forms a structure, can then have some kind of function. And usually that function has to do with interacting with something else. And so in this case, it's a guanine. So this structure right here of the RNA can interact with guanine. If it interacts with guanine, then it forms a different secondary structure. So this is what this red's supposed to be here. This double-stranded piece, in the presence of guanine, this double-stranded piece does not form, and you end up making a transcriptional terminator. Well, if you make a transcriptional terminator, before the RNA polymerase has a chance to make all of these genes for purine biosynthesis, the RNA polymerase falls off. And this, I think, is an incredibly elegant way to regulate because you don't need any extra proteins. It's just the RNA. And the RNA of your transcript can either not be in the presence of guanine, so you need to make guanine. That's what all of these genes are for, purine biosynthesis genes. But in the presence of guanine, you make a terminator, the polymerase falls off, and you can start again. And it turns out that there are ribose switches for lots of different genes, mostly, again, in bacterial systems and mostly binding to small molecules. But you could see this. We looked at the regulation of the trip operon. We looked at the regulation of the lac operon. There you have to be making this whole protein. That protein's got to be interacting with DNA. Here, if you just happen to have an RNA that interacts appropriately with whatever ligand you're looking for, you can regulate here in the absence of any protein whatsoever. So these are ribose switches. This is also known as attenuation. Attenuation is when the RNA polymerase stops before it has a chance to make the rest of the genes that you are looking for. Alternative splicing, we've talked about before in human genes, 75%, 80%, it depends on who you talk to at what time, but large amounts of alternative splicing happen in human genes. So what happens with alternative splicing? Well, usually you use one exon versus another exon. Sometimes it'll get modified. We also talked about this extreme example in Drosophila, where you can have, if I remember correctly, it's about 32,000 different options in terms of how you have splicing. But this splicing is almost always regulated. And how does that regulation work? Well, you've got negative regulation and positive regulation. Negative regulation means you stop using a particular splice site. Positive regulation means that you use a particular splice site that you otherwise wouldn't use. And of course, alternative splicing, one gene can give you lots of different polypeptides. So this is well understood in the case of Drosophila sex determination, which if I could change slides, that would help. Let's see if we can go, move. No, click again. There we go. Fly sex determination, it turns out that flies have exactly the same problem that humans have. You don't want to be expressing too many copies of your sex chromosome. It's XXYY, so it's a slightly different combination of sex chromosomes. But the basic idea is if you have an X to autosome ratio of 1/2, again, just like you would have in male mammals, then you want to develop as a male. However, if you have an X to autosome ratio of 1, i.e. 2 copies, then you want to develop as a female. The way that all of this works is through alternative splicing. All known through Drosophila mutans. And in fact, a lot of this work was done in a lab just downstairs from where I did my PhD at Berkeley. And you would go to happy hour after the seminars, and they'd have all the food set out, and then the students would be going, "That's one of my mutants. It's a curly wing sex comb reduced one." So all the fruit flies floating around were interesting mutants. But that being aside, a little aside there. But again, it's just like you have with the white mutant, the eyeless mutant, etc. If you have a mutation in that particular gene, that's what they name the phenotype. So here, DSX, double sex. Okay, so it means if I have a mutation in the double sex gene, you end up getting sexual characteristics that are both male and female. If you have mutations in the transformer gene, then something that was otherwise going to be male turns out being female and vice versa. And then the sex-lethal gene is if you have mutations in this gene, actually for different reasons, you end up with I think males end up always dying, but females survive. But it's this process. We have the sex-lethal gene product, regulates the transformer gene product, and double sex is the transcriptional regulator, which leads to development of either female or male genes. It's a repressor. So double sex, and you could kind of guess this already. If you lack it, you express both male and female. In the presence of it, you're going to repress either the female genes or the male genes. So in males, you repress female genes. In females, you repress male genes. This is all about the last C-terminal between 100, 150 amino acids or 30 amino acids, which determine where this protein actually binds to DNA. So how do you get alternative C-termini? The way you end up with alternative C-termini is have different splicing. So in males, you don't have use of this cryptic or weak splice site. In females, this splice site is used. How do you get that in females? You actually have a separate splicing regulator, and that's what the sex-lethal regulator is. Sex-lethal doesn't make a splicing regulator in males. It does make a splicing regulator in females. There's a positive feedback loop to make sure that that is maintained. This sex-lethal protein will now block splicing and give you a functional splicing activator. That splicing activator is the one that gives you this extra 30 amino acids here at the end of your protein, female specific. Again, default state here, inactive of all of these, you end up with the male specific double sex. It's only in the females we express these proteins and have a positive feedback loop that leads to alternative splicing and gives you this different tail. So it's not just in flies that you have this different kind of tail formation. You can also, and it turns out this is absolutely required in all of our B cells. You have two different kinds of B cells. You have B cells that make membrane-bound antibodies and B cells that secrete antibodies. These are dependent, again, on a C-terminal part of the protein. If you have polyadenylation that happens here with the membrane-bound proteins, transcription, and again, transcription and messenger RNA processing are happening simultaneously. You transcribe, you make this, there would be a potential cleavage site right here, but this is spliced out. You end up making a poly A tail at this end. This has this green segment. This green segment is highly hydrophobic. This hydrophobic part of your protein sticks in membranes. If, however, you have high amounts of this cleavage stimulation factor present in these activated B cells, this now will bind to this RNA as it's being made by the RNA polymerase, causes cleavage to happen here, poly A tail formation. this now doesn't have a chance to do the splicing and give you this hydrophobic piece. Now you have a hydrophilic piece, and these B cells are secreting antibodies. And this is just literally due to some kind of activation of the immune system. Activation of the immune system, turn up the CSTF. Turning up CSTF, you're going to make poly-A tails earlier on in this particular process. So you can have regulation of splicing, you can have regulation of tailing, and turns out you can also regulate capping. There's less of that happens, but it also makes sense you have less regulation of capping because that's what happens immediately as your messenger RNA is being made. The more bizarre kinds of things that can happen are you can make your RNA, you can splice the RNA, and then, in a few cases, the sequence of that RNA gets changed as well. So what the heck is going on here? So there are a couple of different ones. The main ones that we see in mammals are adenine deamination, and we've kind of talked about this before. This is adenine going to inosine. Where have we seen inosine before? In tRNAs in particular. So tRNAs usually at the first position of the codon, this gives you a wobble base. And so this change of an adenine to an inosine will allow this particular RNA to code for different nucleotides at particular positions. So what does this is the adenine deaminase based on RNA, so the ADAR enzyme. This actually only binds to double-stranded RNA. So what it means is this A right here will only get modified if it's in a double-stranded piece of RNA. Usually that double-stranding piece is right next to an intron. This is absolutely critical for brain development. So if you knock out the ADAR enzyme or you change the sequence here in this intron, you end up not modifying this base and have lots of brain development problems. This was mostly shown in mice, but there are a few human mutations that have some issues with this as well. There's also a protein called ApoBec. I'm not going to get into what the abbreviation is for. It makes no sense whatsoever. This modifies cytosines to uracils. Mostly this seems to be a protective enzyme against RNA viruses. There are lots of RNA viruses that love to infect eukaryotic cells. And this was originally found actually as a defense mechanism against HIV infection. What it does is it changes cytosines to uracils and does a lot of this particularly in the cytoplasm. And those modifications of uracils end up giving you lots of mutations that then end up crashing the virus. There are also modifications that happen in messenger RNA, M6A, so adenine's methylated to the 6th position. It's going to be important for splicing regulation, and 5-methylcytosine now, not in DNA, but in RNA, also is important for probably modifications in the brain. So let's look at one example of RNA editing here. The apolipoprotein B gene normally would have in a non-editing case is CAA. This gets modified to UAA by, again, one of these ApoBec proteins, and this APO from ApoBec is apolipoprotein B, ApoBec. C goes to U, gives you a stop codon. This happens only in intestinal cells, gives you a shorter protein. The longer protein is made in liver cells. So this modification giving you a stop codon, but again, it could also recode your messenger RNA to give you different proteins. So I think we'll talk about export and regulation and CRISPRs on Wednesday. See you then.